today we are going to discuss about Unit 2 of Medicinal Chemistry 1, where we will be discussing the topic SAR of sympathomimetics. First, let us see about how the nervous system works and what are the various types of nervous system. Nervous system in our body can be divided into two categories: central nervous system, peripheral nervous system. Central nervous system is totally controlled by brain and spinal cord. And if we go towards peripheral nervous system, it comprises of somatic nervous system and autonomic nervous system. Somatic nervous system, it will be dealing with voluntary movements of our body, whereas autonomous nervous system, it controls involuntary movements. And autonomous nervous system further divides into sympathetic as well as parasympathetic. So what is a function? Today we'll be dealing about sympathomimetics, means let us see about what are the major functions of a sympathetic nervous system. Sympathetic nervous system majorly deals with increased rate and force of heart contractions, rise in blood pressure, shift of blood flow to skeletal muscles, dilation of bronchioles and pupil, increase in blood glucose level, that is gluconeogenesis. So whenever we are going to deal with sympathomimetics, their functions will also mimic the similar criteria. What are these catecholamines? All these functions of sympathetic nervous system are dealing with various neurotransmitters, and the neurotransmitters which belongs to the sympathetic nervous system are called as catecholamines. These are the one which will be sending the signals from one neuron to the other neuron. So the catecholamines which are involved in sympathetic nervous system are dopamine, epinephrine, norepinephrine. Epinephrine and norepinephrine are nothing but the other name for adrenaline and noradrenaline. Why they are called as catecholamines? The reason for their catecholamines being called is the structure that they will be having. If we see the structure of catecholamines, they would be having this catechol ring in that. So you can see this is the catechol ring. So this ring will be present in all the catecholamines. Maybe few might not be having the hydroxide, that are their derivatives, but most of them, they would be having this catechol ring, and they would be having an amine linkage. So that is why the name we are having that catecholamines. So here we have seen few example in the structures like dopamine having propanethanamine attached to the catechol ring. Similarly, epinephrine, it will be having methyl attached to the aminomethyl and a hydroxyl group on the beta carbon. Okay. Epinephrine, norepinephrine, if we see, it won't be having the methyl, just amine will be there, as well as the beta hydroxyl group will be there on it. So these are catecholamines which will be having the catechol moiety, and to that there will be an amine group attached to it. So that is why these group of neurotransmitters, dopamine, epinephrine, norepinephrine, based on the structure, are regarded as catecholamines. As they are neurotransmitters, they are synthesized in our body. How they are synthesized, let us see. So biosynthesis of catecholamines. So these are synthesized by phenylalanine, that is non-essential amino acid, which is obtained from diet, as well as even it is present in our body. So it is first converted into tyrosine, and then tyrosine get converted into levodopa, and then levodopa gets converts. So here if we see biosynthesis of catecholamines, so in that you can see here, first of all, they'll be having this phenylalanine, which will be either obtained from the diet or in the body, it will be there. So this phenylalanine, by the enzyme phenylalanine hydrolase, this is the enzyme which makes it convert itself into tyrosine. So what is happening here? There is an hydroxyl group that is attaching on the phenylalanine ring, and that is why we call that enzyme, because it is adding the hydroxyl group, OH group, onto the moiety, the phenylalanine moiety. So that is tyrosine. It is getting converted, and then after that, what happened? The tyrosine will get oxidized. Another hydroxyl group will be attached, added up onto it. So we call it as tyrosine oxidase. Okay, so tyrosine oxidase is the enzyme which will be converting the tyrosine into DOPA. What is DOPA? DOPA is actually abbreviation. Levodopa also will tell it as. It is an abbreviation. 3,4 dihydroxy phenylalanine. If we go towards the position, the first, second, third, and fourth, fifth, and the sixth. These are the position of that. So here in phenylalanine, third and fourth position, there are two hydroxy groups. So that is dihydroxy phenylalanine. That is DOPA. That is actually. So this DOPA further under enzyme aromatic L amino acid decarboxylase will be undergoing decarboxylation. The COH moiety which is there, it will get removed now and we will be getting dopamine. The first neurotransmitter that is going to synthesize in our body is dopamine. So here if we require dopamine, so here the stop will occur and they will be releasing the dopamine. But if our body requires other neurotransmitter, norepinephrine, so what happened at that time? Dopamine beta hydroxylase will be converting this dopamine into norepinephrine. So simply what the difference between dopamine and norepinephrine is this hydroxyl group. Only hydroxyl group is extra in that. So that will be added up by the enzyme. Dopamine beta hydroxylase will be adding up that hydroxyl group. Then in presence of the enzyme, if we require epinephrine, adrenaline is required for that instant, not this norepinephrine. So this norepinephrine will get converted into epinephrine by the enzyme PNMT. PNMT, that is the abbreviation will be given for this enzyme. That is phenylethanolamine N-methyl transferase. If you see the difference between norepinephrine and epinephrine is that the CH3 group will be there in epinephrine. That is not there in norepinephrine. Everything is same. Nothing is different. All thing is same. Just a methyl group is extra in that. So that is being transferred by phenylethanolamine and methyl transferase will be providing that methyl to convert norepinephrine into epinephrine. So in this way, in our body, these all three neurotransmitters will be being biosynthesized in our body. So now, after biosynthesis, after performing their action, they have to get eliminated. They need to get metabolized. They need to get removed from our body. We call that as metabolism, as they are getting breaking down into the small molecules. So we call it as catabolism. Okay. So catabolism of catecholamines. So this catabolism of catecholamines, this thing after showing their respective response or the action, there will be two enzymes that are involved for removal of these neurotransmitters to stop their action. That is, one is MAO, monoamine oxidase, and COMT, catechol orthomethyl transferase. So these are the two enzymes which help them to get eliminated or convert them into removable type of enzyme. So this thing happens at the metabolism part that occurs usually in the liver and kidney. So monoamine oxidase will be present in outer membrane of mitochondria and catechol orthomethyl transferase present in cytoplasm. So first is oxidative deamination occur by mono amino oxidase followed by reduction to form 3,4-dihydroxy phenylethylene glycol. So if we see here, so the dopamine, it might be backed by two enzymes, either monoamine oxidase or catechol orthomethyl transferase. So what is the transferring? It is transferring the methyl and it is converting into methoxydopamine. So this methoxydopamine can be easily eliminated. So likewise, if by the action of monoamine oxidase, dopamine will get converted into dihydrophenyl acetic acid. This is also can be easily get eliminated through the liver or kidney. Likewise, if we see epinephrine, it will be converted into normetanephrine. This further gets converted by methylation into metanephrine. Likewise, if we take norepinephrine, it will be converted into metanephrine by catechol O methyl transferase. And both of them, epinephrine as well as norepinephrine, when they react with by the action of monoamine oxidase, they will be getting converted into dihydroxy mandelic acid. So here you can see further detailed explanation has been given here, how they are really getting converted into different. So dopamine by the action of enzyme COMT, catechol orthomethyl transferase, will be converted into 3-methyl oxyglutamine. Okay, and other thing like other enzyme that is monoamine oxidase, it will convert it into 3,4 dihydroxyphenylacetic acid, which on catechol O methyl transferase convert into homovalenic acid. So what is happening here? These all vanillin mandelic acid, if we go towards epinephrine, it will be first reacting with monoamine oxidase here. Then it will convert into 3,4 dihydroxymandelic acid. Then after this 3,4 dihydroxymandelic acid, it will be reacting with the catechol or methyl transferase into vanillin mandelic acid. So these final products, what we are getting, that is homovanillic acid, vanillin mandelic acid, these are the final products that can be easily eliminated through the excretion, so that the effects of these neurotransmitters get stopped or get halted, because after showing their respective response, they need to stop their work. So that is why the catabolism is also important, because it is showing its effect, but it has to stop after a certain period of time. So that is why, like if it is increasing the pressure, blood pressure, after a certain period of time, that blood pressure has to come to normal. So to stop the effect, to reduce make it into back to normal, the metabolism of these neurotransmitters is also important part. Now let us see about structure-activity relationship. So we have seen about what are the neurotransmitters that are involved. So sympathomimetic agents, which we are going to discuss, they will be mimicking the effect of these neurotransmitters. They will be showing similar action. They will be attacking on the same receptors. These neurotransmitters are going to bind to mimic their action. So whenever we are going to come across SAR, like if you go towards full syllabus of medicinal chemistry, it will be having SAR, SAR, SAR again and again. For each category, we need to study the structure-activity relationship. So look here, this is very important concept, and this is fully depend on the mechanism of action. What category does it belong? So structure-activity relationship is a relation between the chemical structure as well as the biological activity. Okay, so in every structure-activity relationship, what we will be doing, we will be taking a prototype compound drug of that category. Then we will be checking out which structure is responsible for its activity, which part of the structure is necessary, what happens if we modify. That is only we will be discussing all in all SAR. What is the importance, which part is important for its activity, what happens if we modify, what happens to activity, whether it increases, decreases, whether the duration of action increases, decreases. So all different in all the different SARs of different categories based on their mechanism of action. Their activity will vary, and based on what the structure is there, the moieties will vary. So if you know what is the structure properly, what are the functional moieties which are present in it, you can write the SAR by your own. So it will be linking the chemical structure to the chemical property and biological activity, including the toxicity with the structure. So now let us see about SAR of sympathomimetic agent. So we are taking here the structure for dopamine, and we can see that they are the three major structural parts that are required to be studied to identify the structure-activity relationship of sympathomimetics. First is the catechol ring. Second is ethylene linkage, which is linking with the amine, and then amino group. These are the three major parts which we need to focus on when we are discussing about sympathomimetic agents. So let us first see that catechol ring substitution is important here. So what happens if different moieties are going to get substituted on catechol ring, and then ethylenic linkage. So which position or where, what sort of moieties can be possible? What happens if there is substitution? What happens if there is no substitution? Then about amino group. Amino group could be having a primary amine, secondary amine, even it could be a tertiary amine. So what happens if the amino group will be having different moieties on it? What happens to the activity? We will be discussing in that category. So first, let us see catechol ring substitution. So 3-hydroxy substitution is essential for alpha activity. So the receptors which are connected with the adrenergic. So sympathomimetics are concerned with adrenergic system. So we will have alpha adrenergic receptors as well as beta adrenergic receptors which are having different activities. Alpha adrenergic receptor has certain activities, beta is having certain activity. So the third hydroxyl group that is on the catechol ring is essential for alpha activity, and 4 hydroxy substitution is essential for beta activity. If you're having catechol ring, it will be having both alpha as well as beta activity. Replacement of catechol ring by resorcinol. Okay, if we are making it resorcinol, what happened? The hydroxyl group will be separated by a point, like if we take catechol, when both the hydroxyl are on side by side, we call it as catechol. When there is a gap of 1 carbon between the hydroxyl group, we call it as resorcinol. So if you are substituting with the resorcinol, so this is resorcinol ring. So if you are substituting the catechol ring with resorcinol, increases beta 2 selectivity and also decreases the metabolism. It cannot be metabolized. Means the duration of action will get enhanced if you are substituting with catechol. But always it is not necessary that we need to have a longer duration of action. But if we are replacing the catechol with resorcinol, it elongates the duration of action. So here it has given the terbitalin in which you are having resorcinol. So it will be having more longer duration of action. And if you are removing the hydroxyl group of catechol, produces alpha selectivity, because third, if you are seeing the third substitution is responsible for alpha selectivity. So if you are removing the 4th hydroxyl, then what happened? Beta selectivity is eliminated. So if you are specifically targeting the alpha receptors to show the response, you will be selecting a compound which is having hydroxyl only on the 3rd position. So example is phenylephrine. This terbitalin is having longer duration of action because it is having resorcinol, long duration of action because of this resorcinol. Now let us see about the next part, that is substitution. What happens if substitution occurs on ethylene linkage? So hydroxyl group substitution on beta carbon is essential for activity. So if you are having a hydroxyl group, OH group on beta carbon, it will be essential, it is necessary. So if you say epinephrine and norepinephrine, adrenaline and noradrenaline will be having this beta hydroxyl group on beta carbon. But substitution of alkyl, methyl or ethyl at alpha decreases the metabolism by monoamine oxidase, produces longer duration of action. Okay, so here if you take example like amphetamine, amphetamine will be having the methyl group attached onto it. So it will be having this CH3 on alpha carbon. So this CH3 on alpha carbon, which will be having on alpha, will be showing the longer duration of reaction of amphetamine. Now let us see what happens. There is a substitution on amino group. if the nitrogen of the amine, it gets substituted with like isoproterenol or isoprenalin, in which you will be having uh, isopropyl ring. This is isopropyl. So isopropyl will be attached here. So isopropyl, if it is there, so there is a moiety it getting attached here. So presence of this isopropyl or any secondary amine, means nitrogen is having an extra substituent onto it. So what happens? Alpha receptor, beta receptor selectivity gets increases, alpha receptor selectivity gets decreases. So primary and secondary amines will have good adrenergic activity. So if you are taking a primary or secondary, will have good adrenergic activity. Now, if we take this adrenergic drugs, that is nothing but our sympathomimetics. These are the medications that will be mimicking. So based on that structure, SAR, what we have discussed, we will be taking different drugs that will be mimicking the action of these neurotransmitters by stimulating the receptors. You have alpha receptors, alpha 1, alpha 2. In beta, we'll be having beta 1, beta 2, beta 3. So these receptors will get activated, and we have seen by the SAR which part, which modification will be affected towards which type of receptors. So based on that, we will be selecting our various sympathomimetics. These are the drugs that will mimic the action of the sympathetic neurotransmitters. So adrenergic related compounds that especially exert their physiological action by stimulating the peripheral sites of sympathetic nervous system. So nerve fiber is a neuron which has a neurotransmitter like adrenaline, noradrenaline, or dopamine will be there. So if you are taking drugs having such structural moiety present in them, that will be attaching onto that part, and it will be mimicking its action. So they will partially mimic action of norepinephrine or epinephrine, and they will be also, look here, they have various names because they are mimicking the action of sympathetic nervous system. They are called as sympathomimetics because they are acting. What is a sympathetic nervous system? It is associated with adrenergic receptors, alpha adrenergic receptors, beta adrenergic receptors. So they are mimicking by attacking or attaching to these receptors. So they are also called as adrenomimetic, or they are doing their action by agonizing the effects on adrenergic nervous system. So they are also called as adrenergic agonist or adrenergic receptor agonist. All these are one and the same category. Sympathomimetics, adrenomimetics, adrenergic agonist, or adrenoreceptor agonist. So these drugs, adrenergic drugs, will stimulate alpha, beta adrenergic receptors. They are specific dopaminergic receptors, and that will be targeting various parts, eyes, heart, and vascular smooth muscles, and they will be showing their action. So here, I'm discussing a couple of drugs here. These drugs are important for your exam point of view because these are star mark drugs. If you take your syllabus copy, there are 2 drugs that are star mark, which means that these drug synthesis is important. You need to write the synthesis. Whenever you see the question paper, whenever they will ask the synthesis, they will ask synthesis only for the star mark drugs. So if you go towards unit 2, you have 2 compounds having the star mark, salbutamol and phenylephrine. So phenylephrine and salbutamol. So that drugs synthesis is very important for your exam point of view. Learn the synthesis and see, like whenever you go towards this adrenergic drugs, keep the structure of dopamine in your mind. What is the difference, modification happening into the other things? If you keep that part in your mind, it will be very easy to study all the drugs. If you see all this sympathomimetic drugs, most of them, like every drug will be have the similar basic structure moiety onto it. Little modifications. What modification is there in salbutamol? You buy 100. What modification is there in epinephrine? You see that. What modification is there in norepinephrine? You see. So if you see this salbutamol, you can see that two major differences are here. Instead of that hydroxyl, we are having methoxy, CH2OH, it is here. This branch is there. As well as you can see this isobutyl, okay, tertiary butyl we are having. So fourth position, we are having two tertiary butyl amino. So if we go towards the numbering here, 1, 2, 3, 4. Because we are giving the IUPAC name through phenol, so we are giving the name in this way. So keep this in mind. So it is on fourth position of phenol, we are having a ethylic linkage. Okay, so on second position of ethyl. Okay, so 1-hydroxy ethyl this is ethyl. So 1-hydroxy ethyl, we are having tertiary butyl amino. Tertiary butyl amino, this is the tertiary butyl amino part. Okay, tertiary butyl amino, 1-hydroxy ethyl, 2-hydroxy methyl. Okay, on second position, what we are having? 2-hydroxy, second of the phenol. Okay, on fourth, we are having this tertiary butyl attached to 1-hydroxy ethyl, 2-hydroxy methyl, phenol. So this is our salbutamol. Okay, so if you know the moieties well, you can by heart the IUPAC name properly and see the changes, difference what we are having extra based on the dopamine, which is the simplest sympathomimetic. So this is our salbutamol. So this salbutamol can be synthesized using 4-hydroxy, 3-hydroxymethyl benzaldehyde. First, it will be in the presence of acetic anhydride. It will be attaching the anhydride moiety, OCH, and then further we will be taking tertiary butyl. Okay, because we need to attach the tertiary butyl here, so that tertiary butyl cyanide. This is cyanide we are taking here. So tertiary butyl cyanide will be helpful for attaching that moiety onto this aldehyde, benzaldehyde. Then later on, in presence of lithium aluminum hydride, these acetoxy will be there. They will get converted into their hydroxyl moiety. So here we will be getting our final salbutamol. Okay. So initially, first we are protecting the hydroxyl groups by the acetylation, by the acetylation, so that the reaction doesn't happen on this position. Then we will be attaching our isobutyl moiety. After addition of isobutyl moiety, deacetylation to give us the final salbutamol. So this salbutamol, if we go towards its mechanism of action, so this is a beta-2 adrenergic receptor stimulant. It is predominant receptor in bronchial smooth muscles, so it will be helping out in bronchodilation and muscle relaxation. So we can guess it can be used for the treatment of asthma because beta-2 receptor is associated with smooth muscle. It will activate adrenaline cyclase, increases cyclic AMP, and smooth muscle relaxation will cause due to which, and then we'll have bronchodilation. So if we go towards the uses of salbutamol, relaxes smooth muscles, lungs, open the airways to improve breathing. So that is why it is used in asthma, chronic bronchitis, emphysema, in which lung that causes shortness of breath, for that also, and to prevent exercise-related asthma. So over-exercise listener. So sometimes it might be if you do a lot of exercise, you might be getting this what is called breathlessness. So it will be used for chronic COPD, like obstructive pulmonary disease, chronic obstructive pulmonary disease and asthma. For that, we will be using this salbutamol. So by the mechanism of action, we can identify where it can be used. Another compound which is important in this MC 2 unit 2, that is phenylephrine. Phenylephrine, you see here, it is not having the catechol group. The main thing you can see, one of this 4-hydroxyl group is not there here. So it is not present on this position. So you can see that is the one part you can keep in mind. So it is having same structure of epinephrine without hydroxyl. So it is 3-hydroxyl on third position. Again, we are dealing with the help of a phenol. So 1, 2, 3. On third position, we are having ethyl, 1 hydroxy, methyl amino ethyl, 1 hydroxy, and 2 methylamine. So on first position of ethanol, we are having hydroxy. And on second position, we are having methylamine. So this is for methylamine. Second position, 2 methylamino. And first position, you are having hydroxy. So on third position of phenol, you are having this whole linkage. Okay, because it is 2 carbon, ethyl for that. Okay, so 3, 1 hydroxy, 2 methyl amino ethyl phenol. So look here, why I'm explaining the IUPAC name, because IUPAC name, if you understand, writing the SARs and all will become very simple. Okay, it's not like if you don't remember, keep in mind, like if you forget to remember by heart the IUPAC name, first you specifically learn the actual name for it, like phenylephrine, this structure that you first identify that, by heart that, then you focus on the IUPAC name, because if you perfectly get the IUPAC name, you will never do the mistake in the structure drawing. Okay, so keep a focus on IUPAC names also. But like in case in exams and all, if you are forgetting the IUPAC name, better not to write the mistaken IUPAC name. What happened? Better not to write the mistake. Just write the journal name if you are forgetting, if you are getting mistake. If you know perfectly, then only you write the IUPAC name. Okay, because unnecessary it will identify that you are doing something wrong if you are having little doubt in the IUPAC name. Okay, so learn with the help of IUPAC name, that will help you to perfectly learn, get the structure. So here, this phenylephrine can be synthesized using 3-chloroacetylphenol. So first what we are doing, we are methylating that 3-chloroacetylphenol with methylamine. Then we'll be getting 1,3-hydroxyphenyl, 2-methylamino ethanol. So we got everything here now. So now catalytic reduction. So we are having ketone here. So we will be simply doing this Clemmensen reduction with zinc amalgam mercury. You can do that. So we will be converting this ketone, simply converting this ketone into its hydroxyl part that will give us the phenylephrine. So simply we can use the catalytic reduction part. So Clemmensen cannot be used because Clemmensen will convert it into ethane. Okay, so that is not applicable. So simply catalytic reduction with the nickel and hydrogen will give us our final product. Yeah. So this is our final product, that is phenylephrine will be obtained. So mechanism of action of phenylephrine, it is selective, direct acting as alpha receptor agonist. It won't have beta. Why it is not having beta? Because it is not having the 4-hydroxy. We have seen it earlier in our SAR that 4-hydroxy is responsible for beta. So because of absent that, it is effective only for alpha. It is potent vasoconstrictor, but less potent than the epinephrine. Activation of alpha causes vasoconstriction of arterioles. So hypotension it might be used. So let us see. So phenylephrine is usually used for relief of stuffy nose, sinus problem, ear symptoms, common cold, flu, allergy, and breathing illness, bronchitis, sinusitis. This medication works by decreasing swelling in the nose, because excess of stuffy nose and all happens due to that dilation of the arterioles and all. So when that is thinning out, it will be decreasing that. It is used as a Medriatic to decrease the eyelids, and it is also used to prolong the action of local anesthetics. So when you go towards this, it will be acting like a prophylactic for local anesthetics. If we see, it will be helping, it's enhancing the action of this local anesthetics. So here, sometimes what happened, these Medriatics are used at the time of surgery, eye surgery. At the time, you need to increases the size of the eye, okay, pupil, so that surgery can be successful. For that, at that time, it is used as Medriatic. Okay, so it has multiple use. So mostly it will use as relieving this nose, stuffy nose, and sinus problems, allergies, and all. Apart from that, it is used for decreasing the swelling of nose and ears. And another important use it is having this Medriatic, specific for beta receptor, it is associated with eye pupil dilation and all. So that is why it is also used as mydriatic. And apart from that, it is also have a use for enhancing the effect of local anesthetics. So let us go towards the quiz for today. Based on what we have discussed till now in this session, like what is the primary mechanism of action of phenylephrine? Beta 2 receptor agonist, selective alpha receptor agonist, non-selective beta receptor agonist, or dopamine receptor agonist. Next, which substitution on catechol ring increases beta 2 selectivity and decreases the metabolism by catechol, like O-methylamine transferase. So whether it is 3-hydroxy substitution, 4-hydroxy, or replacement with the resorcinol ring, or removal of para-hydroxyl group, or 4-hydroxy if you're removing, then you'll get that. Next question is, salbutamol is primarily used for what? Relief of nasal congestion, treating of cardiac arrest, COPD and asthma, treating allergic reactions. Let's go for the fourth question. Which enzyme metabolizes phenylephrine? So COMT, catechol-O-methyl-transferase, esterase, monoamine oxidase, or oxidative deaminase, which will be metabolizing it. Next is substitution of an alkyl group on nitrogen of sympathomimetic affects what? What happens to an alkyl if it is there on nitrogen? Catechol ring activity increases, beta receptor selectivity, dopaminergic receptor stimulation, or duration of metabolism. Next is removal of para-hydroxyl group of catechol ring results. The fourth position hydroxyl, if I remove, what happens? Increase beta 2 selectivity, increase metabolism of COMT, or increase alpha selectivity, or decrease the activity. Next, what is the main use of phenylephrine? Treating bronchospasm, relief of nasal congestion, dopamine receptor agonist, treating heart failure. And next, salbutamol, let us see. A question about salbutamol. Salbutamol acts by stimulating alpha receptor in smooth muscles, beta 2 adrenergic receptor in bronchial smooth muscles, or dopaminergic receptor in cardiac tissue, beta 1 adrenergic receptor in heart muscles. Next is sympathomimetic agent. So which of the following reduces metabolism by monoamine oxidase in sympathomimetic? Substitution of alkyl group in beta carbon, substitution of alkyl group at alpha carbon, presence of catechol moiety, or removal of 4-hydroxyl group. Coming to the last question, which of the following statement about SAR of sympathomimetic agent is true? Substitution of M-hydroxyl group increases alpha selectivity. 3,4 dihydroxy substitution is required for both alpha and beta activity. Hydroxy substitution of alpha is essential for activity. Replacing the catechol with resorcinol decreases beta 2 selectivity. Means all these 4 are having closely related, but only one is specifically correct. So which one among this is correct? So this is about the MCQ based on our today's presentation. So if you go towards this second chapter, in that we have many drugs are there: direct acting sympathomimetics, indirect acting sympathomimetics, mixed acting sympathomimetics are there in that. So you need to study all of them, but these two which I have discussed, their synthesis is important in exam. So they will be asking directly about the synthesis of these two drugs. Okay, so the questions that could be coming from this second unit mostly includes draw the following structures: salbutamol, propranolol. This comes under beta antagonist. Okay, beta adrenergic antagonist, propranolol, atenolol, they comes under that. So they might be simply asking for 2 marks or 3 marks, very short question, simply asking 3 drugs and their 3 structures. Okay. So next is explain the catabolism of catecholamines. Just now we have discussed that catabolism can become for 3 to 5 marks, sometimes it might come. Explain SAR of adrenergic agonist. So what we have discussed today, sympathomimetics are nothing but adrenergic agonist or adrenomimetics. So explain the SAR of beta blockers. In that you will be taking propranolol as prototype drug, and we will be discussing the beta blockers SAR there. And next question is there like synthesis and mechanism action and uses of phenylephrine, because this is among the important star mark drug, phenylephrine and salbutamol. For them, they might be asking the synthesis, mechanism action and uses. So these two from this second unit, you have again in antagonist, again beta antagonist, alpha antagonist. Again, you have a couple of drugs in which you will be having star mark for them. You need to study the synthesis for them. And next, write the name of the adrenergic receptor and their distribution. If you go towards adrenergic receptor, before discussing that, we have alpha, beta, and that again alpha 1, alpha 2, beta 1, beta 2, beta 3. Okay, so that we haven't discussed in today because we need to discuss about these concept about SAR and catabolism and biosynthesis of amines. So we need to have an idea about that different receptors that will help you a lot by understanding the mechanism of action. Because if you know the mechanism of action, indirectly you can predict the uses also, because it is acting on that, so it is useful for that purpose. So if you go towards the long answer questions that is going to be asking in this is like explain the biosynthesis of catecholamines along with the reaction. So we will be really asking about biosynthesis. So first write that how they will be synthesized using a phenylalanine, how it is getting converted into dopa, tyrosine first, then to dopa, then dopa to dopamine, dopamine to noradrenaline, noradrenaline to adrenaline. Next, SAR of sympathomimetic. Just now we have discussed, so that SAR will be asked. Next, synthesis and uses of salbutamol and phenylephrine. Individually, they might be asking. If they are asking for long answer, they would be asking together. But mostly, individually, one by one, they might be asking the synthesis of salbutamol and synthesis of phenylephrine. So that's about this today's session. So in which we have discussed a lot of concepts, like we have seen very important 4 concepts we have seen to be specific. So first of all, we have seen about what are sympathomimetic agents. We have seen in general intro regarding to that. Then we have seen about what are these sympathomimetic agents. SAR we have discussed structure activity relationship. Before going to that, we have discussed about biosynthesis of catecholamines and catabolism of catecholamines. These are the 3 concepts. And the 4th was about synthesis of important drugs, like 2 important drugs of the second unit, that is salbutamol synthesis as well as phenylephrine synthesis. So for much more detail about this medicinal chemistry, you need to follow up this app so that you can be following, subscribe to the channel of Prakara. That will be helping out to know more about different subjects. Also, you will be having these webinar sessions, as well as if you go to the app and buy the app, you will be getting full detailed information about whole chapters, full detailed chapters of each and every concept will be discussed by the professionals, and they will be explaining it full in detail about each concept with the full-fledged notes. You will be getting full-fledged notes what I have discussed just now, and that you will be having full detailed notes on each and every topic of different subjects. So to get more and more information regarding the pharmacy related subjects, follow up this app, subscribe to this channel so that you will be updated regarding various webinars on different topics. And to much get into detail about the subject, you follow the app, buy the app. In that you can buy the different modules. So buy the modules in which you will be able to access whole notes and everything in that. So that's it for today.